So I'd just like to introduce myself. So I'm Phil Atherton from the University of Nottingham, and the task today has been to describe some of the aspects around the development and application of stable isotope tracers to exercise physiology. And for many of the examples that I'm going to provide, some of the sort of tracer techniques that I'll describe have been sort of well used over the years, and maybe some people will be familiar with these and maybe some won't. And what I'd like to do as well is just cover some areas of sort of emerging interest around stable isotopes. Some of these applications to date haven't necessarily involved direct application to exercise as yet, but I think that they have significant potential to do so. So first of all, I'd just like to describe a little bit more about what stable isotopes are. So, because I guess that many of the audience might not be familiar with what these things are. So essentially a stable isotope is a sort of natural occurring atom. So for example, carbon, oxygen, nitrogen. But in these stable isotopes, they actually contain extra neutrons. So you can actually see as an example here, if I can actually get this thing to work, of a hydrogen and deuterium. So what you can see here, this is a hydrogen atom here, and you've got one proton. And what happens is if you get this extra neutron within that nucleus here of the atom, it actually becomes a compound called deuterium. So deuterium is, in essence, here, as you can see, is actually the stable isotope, the rare stable isotope of hydrogen. So this gives just an example, really, of what an isotope actually is. And one of the important features about isotopes is that they behave identically. So even though, from a chemical perspective, even though these things actually differ in the sort of number of protons, they actually behave chemically very, very similarly. So that means that if we utilize them to trace metabolism, using whatever sort of approach we choose, and I'll detail some of these in forthcoming slides, and what we're able to do is actually trace and follow these particular rare isotopes into the chemistries that we're interested in. In essence, that's why they're called stable isotopes. The other key feature about these that people often must understand is that these stable isotopes are stable in that they're non-radioactive. And that basically means that they don't undergo decay. So whilst radioactive isotopes will undergo decay and can create things like DNA damage and ionizing radiation and such like, these stable isotopes are extremely stable and they remain in a non-decaying state, which is really, really important. And that basically means that they're safe for use in humans. And basically, what the key element of the difference here, the key sort of application of these stable isotopes to exercise physiology and many others, is that essentially we can actually detect, using mass spectrometry typically, the rare isotopes and the ratio between the rare isotopes and the common isotopes. And in examples that I'll provide, this gives you techniques and technologies by which you can actually confer different things around what's happening with metabolism. And this is just another example here of a carbon atom here. And essentially what you can see is the most common form of carbon is this carbon-12 here, whereas carbon-13 makes up around about only 1% of all the carbon. And essentially what you can see is the 12 is delineated by the fact that there would be 6 protons and 6 neutrons, making the atomic weight to be 12. However, when you add this extra neutron, this makes it carbon-13. So many of the traces, for example, that I'll describe today would either be, for example, the sort of deuterium form of hydrogen, or, for example, the stable isotope of carbon. But as you can see, there are lots of different isotopic variances of these key chemical elements. So there are many, many applications, and obviously this is a relatively brief talk and introduction to this, but I just want to go over some of the sort of commonly used applications of stable isotopes. So in exercise physiology in particular, one of the most common ones in the literature is the measure of direct incorporation of these isotopes into polymers. So for example, there's been a huge amount of work looking at using amino acid tracers and their incorporation into protein, and you'll hear about this, no doubt, during this conference. So, for example, you can see here you've got various different types of tracers. Here you've got a leucine amino acid that's labelled, so 2 of the carbons are labelled with this 13C element, 13C6, where 6 of the carbons are labelled with this 13C. But you can also see, actually, that you can also get sort of 15N labelled proline, for example. So, for example, if you were interested in looking at incorporation into collagen molecules or extracellular matrix, which are very rich in non-essential amino acids, such as proline, then this could be, for example, your tracer of choice. The other things that you can do, you can look at lots of different substrates if you have the specific traces to do so. So you can also look, for example, at using sort of tritiated or deuterated glucose and its incorporation into glycogen. Of course, you can use fatty acid traces such as palmitate, 13C labeled or universally labeled palmitate, and you can actually look at their incorporation into triglycerides. But also, depending on the tracer, you can actually, in theory, follow the synthesis of DNA and RNA by incorporating traces into the bases that make them up, either the ribose or the deoxyribose. And one tracer methodology that's come to the fore more recently that I'll describe a little bit more about is one called D2O, or heavy water, in which two of the hydrogens on water are replaced with deuterium. And essentially, what we can do with this tracer is, rather than having a substrate-specific tracer where you have to, for example, have an amino acid tracer to trace protein and a fatty acid tracer to trace triglyceride synthesis, well, actually, potentially, what you can do with D2O and heavy water is actually measure and label all at the same time protein, lipids, and DNA. So that arrives at a lot of potential for measurement of substrate turnover. What you can also do, and again, which is common in the literature and what's been used in lots of exercise studies, is with appropriate sampling and blood flow, you can actually measure arteriovenous balance. And people may have come across this quite simply, for example, at looking at arteriovenous sampling and measuring glucose in the artery and the vein, and therefore being able to look at sort of AV difference in glucose as a proxy, for example, for glucose uptake across a limb. And in order to do this, you actually need to quantify blood flow, because you need to know what the flow is, because that will also contribute to the AV difference. So with appropriate blood sampling, it's also possible by infusion of these tracers, such as amino acid tracers, that you can measure the synthesis and also the release and intermediary metabolism of labeled compounds. And essentially, I'll describe this. I'll just show this as a schematic in the next slide. But what you do when you infuse these tracers is you're able to actually look at the rate of so-called disappearance, or RD, and the rate of appearance, or RA, which in effect equates to the synthesis and breakdown. And importantly, if you use an amino acid such as phenylalanine and you're interested in skeletal muscle, phenylalanine, not being a branch chain, is not metabolized in muscle, which is important if you're looking at dilution across an artery and a vein. So this method basically allows you, by infusion of these tracers, to look at AV difference and AV balance across a limb. You can also measure oxidation, energy expenditure, and metabolic flux through pathways using tracers. So just to provide some examples here, if you're interested during exercise, for example, about what substrates are being used, and you want to go beyond, for example, indirect calorimetry, looking at VO2 and VCO2, what you can actually do is, again, provide tracers which have this carbon labeled motif. And at the terminal end of metabolism, what essentially happens is that this CO2 that's expired has the 13CO2 motif. So basically, if you quantify in the breath, you can actually quantify the 13CO2 enrichment. And if you're giving, for example, an amino acid tracer, you'd be able to look at the contribution of amino acids to energy production given a specific, say, exercise stimulus. Using another doubly labeled water technique is which is actually the gold standard in order to confer energy expenditure by looking at dilution of these different traces in the breath and in the urine. But also at the same time, there's really quite novel techniques and technologies that have come about, and I've given a few examples of these in later slides. So by providing these tracers, again, as these tracers undergo sort of incorporate into substrates and undergo catabolism within tissues, what you can actually do is track the flux of these tracers. So you can actually look whereabouts the labels of these tracers appear in downstream metabolites, and therefore get an idea of what the fate of a substrate is given a particular stimulus. And then just finally on this, there's actually some new methods, some relatively minimally invasive methods by which you can now establish a whole body protein muscle breakdown and also muscle mass just using tracers. So this is really just a sort of a general overview of what the possibilities are. And as I'll tell you at the end of this, we're actually hosting a Derby tracer course in Derby, which will go into much greater detail for anyone interested in these techniques and technologies. So this is just a sort of overview schematic really of what I described previously, in that this is a standard methodology that's been used in order to arrive at protein synthesis and protein breakdown in respect of many exercise and nutrition-related studies. And what you basically can see here is if you just look at the differences in label, you can see here that there's the amino acid tracer in red and the endogenous amino acid in black. Essentially, if you infuse this into a vein, what it does is it gets into the, obviously, into the AV pool. Essentially, then what will happen is these amino acids will reach a tissue. This, for example, is skeletal muscle. They'll transport in the muscle through the amino acid transporters, equilibrate with the intracellular pool, and eventually what will happen is these amino acids behaving exactly the same, these tracer amino acids behave exactly the same as the endogenous ones, and what they can do is, of course, bind and charge the cognate tRNA, and then they can incorporate into peptides. And by simply measuring the enrichment of the tracer within these peptides, by taking a small tissue sample, we're able to calculate protein synthesis. Similarly, in terms of breakdown, some of these, of course, will then leave, some of these amino acids will, of course, leave the cell and will end up into the venous circulation. And basically, simply by looking at the enrichment in the tracer in the artery and the vein, we're able to arrive at the rate of disappearance, rate of disappearance of the tracer, which equates to synthesis, and the rate of appearance into the venous pool, which equates to breakdown. So essentially, these methodologies have provided us a way in which we can quantify both synthesis and breakdown. And basically, this is what, for anyone unfamiliar with these techniques, this is what a standard protocol would look like. So you would have a period, what we would typically have is 2 periods in a basic study design. You'd have a baseline period here, and you'd have an intervention period here. And what you would do is infuse a tracer here and take AV blood samples if you're interested in quantifying both synthesis, breakdown, and arriving at net balance. And of course, for taking biopsies in order to measure the direct incorporation of the tracer into muscle protein. And essentially, what this has done is provided a host of information which is beyond the scope of this talk, a little bit about sort of what we've become to learn about how exercise and nutrition in particular modulates muscle metabolism. So, for example, what have these tracer methods taught us? Well, they've taught us, and again, I'm sure you'll see aspects of this in some of the talks later in this conference. For example, they've taught us that certain exercises are associated with fraction-specific responses. That means that if you're able to actually subfraction a biopsy into, say, mitochondria or collagen fractions or myofibrillar fractions, under certain circumstances, you get specific stimulations of one fraction over another. So you're actually able to see how different exercises modulate different fractions. It's also helped us optimize exercise protocols. So, for example, being able to actually determine what is the best way, what is the best sort of resistance exercise to maximize protein synthesis acutely. And this has basically led us to be able to make some recommendations about how that may confer greater trainability if these exercises were to be sustained. Then there's been a bunch around sort of exercise and nutrient. This has made up a huge sort of part of research in this area using these isotopes. So, for example, how much protein should you have to maximize the exercise response? What other nutraceuticals might work? So, for example, some particular compounds that may be purported to have an ergogenic effect. You can, of course, measure whether or not they are able to potentiate any response to exercise. The timing of nutrition as well, and of course the composition of macronutrients in particular, but also other factors such as the effect of training status. So how does actually being trained impact your ability to respond? And of course, beyond the exercise per se, what is the impact of age and diseases on these anabolic and catabolic processes? And also, of course, how different modes of exercise actually modulate protein turnover. So these methods have provided a lot of information around exercise physiology. But one of the limitations to these methodologies is basically that they require that the individual undergoing the study, it has to be pretty much a captive audience. They have to basically be led in a bed or certainly be within a controlled laboratory environment throughout the period of study. And essentially, this is because for most of these types of studies, it requires that there's a sterile infusion of these specific tracers into a vein. That means there's a sort of continuous infusion. That means, of course, that it's very limited about what you can do with people during these particular studies. So you might be able to do a single bout of exercise, for example, but if they've got lines going in, it becomes very, very challenging to do anything significantly beyond that. These measures are typically restricted to less than 24 hours, again, for the same reasons, in that you require these constant infusions, and therefore you're pretty limited as to how long you can undergo these procedures for. And the other limitation really is that a single tracer is required for every substrate of interest. So if you have a particular substrate of interest, so fat or carbohydrate or protein, for example, you have to have a tracer for each one of these independently. And of course, there's sort of a cost element as well. So one of the earliest tracers that was actually introduced in the 1930s, I think, was one called deuterium oxide. And I mentioned this before, where two of the hydrogens on water are deuteriums. And basically, the benefits of this tracer, or potential benefits, was that it's orally administered, so you can just drink it. You don't need to provide an infusion. You can actually study people in a free living environment, so they can go home and go about their daily business while you study them. You can measure for chronic periods. You can do hours, in theory, days and weeks. So that means that gives a lot of scope for interventions. And also, it labels multiple substrates through mechanisms I'll show briefly in the next slide. And actually, depending upon the dosing strategy you provide, it actually reduces the cost significantly as well. So here's a sort of overview slide, a summary of the sort of potential for D2O in terms of being able to quantify aspects of metabolism. So you can see here, basically, there are three major areas of interest that I've sort of highlighted. One is around about DNA synthesis here. One, of course, is around about protein synthesis here. And one is what I've termed muscle quality, but actually I'm mainly actually talking there about lipid synthesis within tissue and potentially ectopic fat deposition. So what you can see basically here is that once you take D2O, it rapidly equilibrates with the body water pool. And then this D2O, what will happen is the deuteriums will actually exchange with lots and lots of substrates. But as you can see, suffice it to say that for here, that D2O here will actually label up eventually some of the deoxyribose in DNA. It will label up many of the non-essential amino acids through transamination. So alanine is shown here. And also at the same time, you can actually, it will label through fatty acid processes, synthesis processes, triglycerides. And this means essentially that we're able to arrive at measures of protein synthesis, DNA synthesis, and in theory, lipid synthesis as well. So I'm just going to give some examples here of some validations of this technique that we've performed in our lab, for example. So we did two studies basically here. I'll just summarize these very quickly. I showed what these sort of acute studies looked like before. So what our first study was aimed to do was to determine whether or not we were able to prove that the D2O tracer performed in a similar way to the carbon amino acid tracers in terms of tracking protein synthesis. So this is the first study we did on the top, in which we basically gave people a 20-gram, I think it was, bolus of essential amino acids, which will stimulate protein synthesis maximally. And we gave them either 13C6 phenylalanine, or we gave them a single bolus of deuterium oxide. In the second study here, we did a study for 8 days. And in this study, what we did was we did unilateral exercise. So we basically trained one of their legs. We gave them a bolus of D2O here. And then what we did is we had one rest leg and one exercise leg. And we exercised them about every day 0, day 2, day 4, and day 8. And basically, we measured the protein synthesis response over time. And what we were looking for was to see whether or not they reliably gave us information in terms of what we would expect to be the absolute FSR, fractional synthesis rate, but also at the same time whether or not we could actually separate the anabolic responses. So this is what we found when we did this. This was our feeding study. This is the saliva enrichment, which is the proxy for body water enrichment of deuterium. And this was the same for the plasma 13C6, so the precursors. And then what we measured was fractional synthesis rates and muscle synthesis here. And you can see that this is the stimulation in response to feeding here. And when we gave D2O, we actually found that the stimulation was recapitulated, suggesting that the tracer and the techniques and technologies that we applied were able to be used for oral administered D2O in order to quantify protein synthesis. And when we did some Band-Altman plots, we found that actually the methods performed relatively equally, meaning that this tracer did seem to have efficacy for measuring acute MPS in humans. We then did this 8-day study, which was this unilateral exercise. And what we found, this is the body water enrichment. We remember we gave a single bolus here, so you can see this exponential decay in body water enrichment of the deuterium. If you then look at the alanine-bound, the deuterium-bound protein here, what you can see here, this is the exercise, the solid line, and the non-exercise. You can see over time, cumulatively, we were able to actually distinguish between the exercise and the non-exercise leg. suggesting that, again, we're actually using this longer-term method. We're actually able to determine an anabolic effect over a longer period in the exercise versus the control leg. When we looked at myofibrillar protein synthesis, again, this is the exercise leg and this is the non-exercise leg. And basically, these are just split into different bins. What you're able to do is calculate either 0 to 2, 0 to 4, 0 to 8, or you can actually do them between each of those periods. But suffice it to say that what we could see was that the exercise leg remained sustainably above that of the non-exercise leg over this period of time. And what was really important about this as well is that what we found was that rates of synthesis were equivalent to amino acid traces which have been acquired, of course, over periods of hours, such that our turnover rates equated to around about 1.2 to 1.5% per day. One of the reasons why it's very important to take this into a chronic setting beyond the obvious is because what we found in the past, and this was a paper in collaboration with Stu Phillips in this audience, here is that if you actually measure the acute response to exercise under fed conditions and then see how that relates to hypertrophy, what you find here is that the FSR, the fractional synthesis rate here, and compared to MRI-mediated muscle volume change, does not at all well correlate, suggesting that the acute exercise bout does not necessarily confer what happens with regard to muscle hypertrophy 4, 6, 8, 12 weeks later or whatever. However, when we actually look at the D2O and do a chronic study, so this is a 6-week training study, what we find is that over the first few weeks, you get this robust increase in protein synthesis. Remember, this is per day, so actually the amount of change is actually reasonably significant over those 3 weeks of training. And what we also find is that when we look at muscle thickness and how it correlates to fractional synthesis rate, we actually see a much better and much improved efficacy to determine whether or not the cumulative effects of the resistance exercise are correlating with that of muscle hypertrophy, suggesting that the slightly longer term measures will be a much better readout for ensuring hypertrophy. By doing this and using the D2O method as well at the same time as quantifying muscle mass, you're also able to extrapolate a number of other things. So for example, here I've shown that you're actually able to calculate the absolute synthesis rate. So if you know how much muscle somebody has gained and you know what the fractional synthetic rate is over a period of time, what you're able to do is multiply them and calculate basically how many grams a day of protein is actually being made within the muscle. And if you can calculate, you know the muscle mass and you know the absolute synthesis rate, you can also calculate the absolute breakdown rate, and you can determine whether or not, or make some inference about the particular impact of, say, any sort of exercise or other intervention upon both absolute synthesis rate and absolute breakdown rate of muscle protein. One of the other important aspects I think I mentioned before was that what these methods give you the ability to do is look at different fractions within the tissues. So for example, what I showed in the first slide, in one of the earlier slides about the 8-day study, is that we measured myofibrillar protein synthesis. However, we also in that study measure collagen and sarcoplasmic FSR, for example. And these, of course, require very careful extraction to ensure that you have a proper good fraction purity. And what you can see here is, again, that you're able to see that collagen synthesis was also stimulated sustainably over time during this particular exercise intervention. And this is a study which supports the notion of using deuterium oxide, again, not from our lab, from Ben Miller, one of Ben Miller's studies, basically showing that in 4 weeks of sprint interval training, when comparing males and females, the male responses in each of these fractions, mitochondrial, mixed, and sarcoplasmic, was greater in the males than that of the females. So this can give you quite a lot of insight about how exercise is regulating specific fractions. Taking that a step further, we used an animal model of trainability in which a selective outbreeding program has been developed to generate animals which are either exhibit low or high response traits to endurance exercise. And what we did was we looked at these fraction-specific responses in these animals when we exposed them to the exercise response. And what you can see here, essentially, the main finding we got was that the low responders, for example, show a decline in both mitochondrial and myofibrillar fractional synthesis rate across the range of the training program per se, suggesting that these should be in some way directly linked to mitochondrial biogenesis. One other thing I mentioned is that, of course, one of the benefits of D2O is that you don't necessarily have to be limited to things like proteins. You can actually look at other substrates as well. And one of great interest, particularly around things like hypertrophy, is the role of satellite cells or satellite cells in muscle growth. And of course, this remains a relatively contentious issue, depending on which literature you read. But this is, again, another study from Ben Miller's lab showing essentially that when exposed to a training program, although the groups were a bit random in that there was a young sedentary group compared to an old exercise group, in the young sedentary group, they were unable to detect any significant change in DNA synthesis under rested conditions, which is consistent with the notion that DNA turnover in adult muscle is relatively low, of course. However, in the older exercise group, they were able to see that there was an increase, a significant increase in new DNA that was synthesized, suggesting that this DNA is coming about through increased satellite cell activation and incorporation of their nuclei into the existing fibers. And what they also found in this is that the muscle protein synthesis response was actually correlated to the percent of new DNA, suggesting that there was some link between the capacity to increase synthesis and thereafter the capacity to increase DNA content as well. One of the other things that you can do with D2O, beyond simply looking at either gross FSR of muscle, you can actually also look at specific proteins per se. So you can go from looking at whole sort of muscle like mixed lysates, you can look at specific fractions, but actually if you combine this approach with proteomics, what you can also do is look at individual proteins. So here's an example of how this has been used. This is an animal study. This is the days of labeling. They've given a beta agonist here, clenbuterol, and you can see here that over time, the deuterium labeling in the gastrocnemius muscle is actually greater when given this particular drug, which isn't surprising. What they also found, which was really interesting, is that they were able to actually look in the plasma, go to the plasma of these animals, and basically immunoprecipitate out muscle-specific proteins and measure their synthesis. So here's CK, creatine kinase, muscle-specific. So around about 90% of this particular CK is expressed principally in skeletal muscle. And when they looked at the correlation between the synthesis rate of creatine kinase in muscle and indeed the mixed muscle synthesis and that of the plasma, what they found was a very nice correlation, suggesting that actually under certain circumstances, you can actually use D2O in order to generate a so-called virtual biopsy in order to extrapolate what's happening in the muscle by looking at muscle-specific proteins. And they took that a step further by actually looking at a load in response to a sprint interval training in humans, and they looked at FSR of different individual proteins. So they do a typical tryptic digest in order to isolate the proteins, do a tryptic digest, but then not just identify the proteins, but identify the enrichment of deuterium in these particular peptides. And what that allows you to do is generate a list, basically, of proteins and a calculation and extrapolation of their synthesis rates. And what they found, basically, was a number of proteins here, just as an example, that were induced in response to sprint interval training. And you can do much the same as you do with sort of genome biology. You can actually look at ontologies. And interestingly, they found, as you'd probably expect, that the glucose metabolic process or proteins regulating anaerobic metabolism were actually upregulated during this training response. So just a few important considerations before D2Oing it. So first of all, you do have to consider whether how you're going to do the dosing. Again, there'll be a lot more detail on this for anyone that's interested, but you have to consider, do you give a single bolus of D2O? Do you try and create some kind of pseudo-steady state by giving regular top-ups? Or do you try and engender a situation of a true steady state where you give sufficient D2O with enough regularity that the body water pool remains enriched on at a steady level? You can also measure tissue and fraction turnover, as I've shown you, and also individual proteins. And the other thing that you consider is whether you do constant measurements over an entire block of training, or maybe if you do a long-term intervention, you could do a sort of window at the beginning, the middle, and the end. And one other thing that's important to state is that the dosing depends of D2O. The absolute dosing depends on the sensitivity of the mass spectrometry that you actually apply. So just a couple of final bits, really, about some of the additional... is there time for this, Paul?
Not really.
All right, I'll stop then. It's always the way. So in that case, I'll fast forward. So just some food for thought, really. Sorry, I had to miss that last bit. I always talk too long. Stable isotope traces are still being used for traditional metabolic approaches such as substrate turnover and oxidation. What hopefully I've shown you today is that D2O offers the opportunity to combine protein, DNA, and even triglyceride turnover, hopefully minimally invasively and also in a chronic situation, which has not been easily possible before. I've shown you a little bit of it about the fact that combining traces with omics approaches can yield information such as, for example, the metabolic fate of substrates and individual protein turnover on an individual level. And of course, this gives you additional possibilities to probe systemic proteins that that putatively has leaked out of skeletal muscle, for example, during exercise. I won't mention this because I've not talked about it now. But I would say, just conclude by saying combining multiple isotopes to multiple endpoints is very, very powerful. So we've got now so many tools by which we can actually combine tracer approaches and tracers that we can get a huge amount of information about what is actually happening in vivo, which I think it should not be underestimated. And I think that stable isotopes, is hopefully you can see a snapshot of this, really do facilitate true dynamic mechanistic insight in humans or in vivo in animal models. So just a final point really is a plug. I mentioned before that we've got this ESPN intensive course in tracer methodology and metabolism, and anyone that thinks they might want to apply these methodologies to their research might want to consider attending this. It's in Derby, on Derby campus at Nottingham, at the 11th and 12th of June, and further information is available. So with that, I'll end. Thank you.
Okay, thank you.